Hello, and welcome to our podcast series on the specific immune system, also called the adaptive immune system. This is talking about the types of antibodies. So, the previous podcast was on structures of antibodies, and this one will talk about the different types of antibodies. So, if you look at the right part of the cartoon, you can see a typical antibody with its two heavy chains bound together by disulfide bonds. Both heavy chains have a constant region and a variable region. And then there are two light chains, and the light chains also have a constant region and a variable region. Where the variable region of the heavy chain and the variable region of the light chain form a pocket, that's the antigen/epitope binding site. And then the antibody in general is in the shape of a Y, so you have two arms of that Y, and that's called the FAB region. And then you have the stem of the Y, that's the FC region, or the stem of the antibody. So there's 5 basic types of FC regions, or there's 5 basic types of stems. There's mu, gamma, alpha, epsilon, and delta. So the plasma cells, those are B cells that found an antigen/epitope with their B cell receptors. They bound to the antigen epitope, and then those B cells became activated, and then they were called plasma cells. Plasma cells can make the antibodies, and those antibodies have the same antigen binding sites that the original B cell receptors had in the B cell. So if you are a plasma cell, you're making antibodies. Antibodies are proteins. That means that antibodies have to be made from a gene template. And plasma cells can take the gene that makes the variable region, and it can use a basic stem, but then it can attach different antigen binding sites to that common stem. And those stems form the 5 classes of antibodies, M, A, D, G, and E. So when you're sick and your body is responding to a foreign invader, and you begin to make your antibodies against that foreign invader, or you begin to make your immunoglobulins, which is just another word for antibody, then IgM is the first type of antibody that's made during that initial phase of the infection or immune response. So this is a cartoon of IgM. They call it a pentamer because it's got 5 monomers, or single unit antibodies, linked together in a J region. So that green ball in the cartoon is not a pathogen. It's just the part where the 5 different monomeric antibodies in an M-type antibody are joined together. The next antibody that you would make in your immune response to that foreign invader is IgG, and this is a single unit, so it's a monomer. It's not a pentamer like IgM, but IgG is the most common antibody. It's the longest lasting antibody, so that means it doesn't degrade. And then IgG is also the only antibody that will cross the placenta. So IgG can really easily leave the blood vessel in the capillary, or leave that part of the blood vessel. It's called a capillary. IgG can leave at the capillary. It can go into extracellular spaces, and that's oftentimes where pathogens are hiding, so IgG can find them. IgA is the next class of antibody that we're going to talk about. IgA can be a monomer. It can also be a dimer. So if you look in the cartoon, this is the dimeric form of IgA. So you've got two antibodies joined in a J chain at their stem regions. And then there's a secretory component, because IgA is the antibody that's associated with secretions like secretions from your lacrimal glands, so tears, and also secretions from breast milk. The next class of antibody is IgE, so that's a monomer. And IgE tends to be upregulated when you have a parasitic worm infection. So you'll make a lot of IgE, and then eosinophils can bind to that IgE, and then the eosinophils, they can secrete toxins to kill the parasitic worm. Finally, there's the last class of antibodies is IgD, and they're just a membrane-bound antigen receptor. So remember, the antigen binding site on the antibodies looking for foreign antigens, so they can find the foreign invaders. And they usually find IgD is in B cells as a B cell receptor.